Okay, so today we will be starting with the lecture Staphylococcus species. This is the topic of veterinary microbiology of the chapter of the unit pathogenic bacteria. So these are my short notes which are made from the book of Quinn and Carter. So it is a standard textbook for veterinary microbiology. So let us start. So Staphylococcus are gram positive. So I will upload a lecture later on what is gram positive, what is gram negative. So first till now, for now we can learn that it's a gram positive bacteria. It is cocci, as the name suggests, Staphylococcus. So it is a cocci or spherical. Its diameter is 1 micrometer. This data is also important. This point is extremely important. Third point, form bunches of grapes. So it forms irregular cluster resembling bunches of grapes. So this point will be asked in the question, but practically also this will be asked in, or this will be important when studying parasitology in third year. Okay. Now habitat, commensals on skin and mucous membranes, and opportunistic pathogen causing pyogenic infection. So commensals means a type of commensals can be thought of closely similar to symbiotic relationship, and commensals on skin and mucous membranes. And opportunistic pathogen causing pyogenic infection. Pyogenic infection means pus forming infection. Okay, these are non-motile, thus they do not contain flagella. Catalase positive or oxidase negative. So these are catalase positive or oxidase negative. So these catalase and oxidase are the type of biochemical test. So catalase positive means that the Staphylococcus produces enzymes catalase, and which catalase enzyme converts H2O2 into H2O and O2. So why is the need for there any catalase enzyme? The need for catalase enzyme is because H2O2 is a free radical in itself. So just to prevent the bacteria from the damage of the free radical H2O2, this catalase enzyme is present. Okay. The oxidase negative means oxidase, it does not have any enzyme called cytochrome C oxidase. So it will not perform the functions of cytochrome C oxidase because it does not have that enzyme. Okay. Next is Staphylococcus. Okay, this Staphylococcus is generally found in those areas of the body of animal which are considered sterile. Animal and human both. These are found in those areas of the body which are considered sterile. For example, synovial fluid in the joints and CSF, cerebrospinal fluid in brain and spinal cord. Okay. Now, or also in the artificial media, staph are isolated from extremely pure cultures. So these are found in those areas which are extremely pure or sterile. Okay. Now this point is important, characteristics. So characteristics of Staphylococcus. So first is pigmentation, second is hemolysis. So pigmentation, its colonies are white and opaque. The colonies of Staphylococcus species is white and opaque, and diameter is 4 millimeter. But the colonies of Staph aureus, which infects bovines and humans, are golden yellow. Okay, so we learned that the colonies of Staphylococcus is white and opaque. Colony of Staphylococcus is white and opaque, but the colonies formed by Staphylococcus aureus, the species of Staphylococcus, subspecies of Staphylococcus aureus, which infects bovines and humans, those colonies are golden yellow. Why? Because of the presence of a pigment called staphyloxanthin. That is not written here, but I have learned that there is a pigment called staphyloxanthin which is produced by S. aureus, subspecies of S. aureus, which infects bovines and humans, and that staphyloxanthin gives the colony a golden yellow color. And that staphyloxanthin is not only present for the color, it performs various specific and important functions important for that bacteria, Staphylococcus subspecies. So for example, it will provide them ability to increase their virulence, and it will also give them the ability to protect themselves from the free radicals or reactive species produced by the normal cell. Okay. So first was pigmentation, second is hemolysis. So hemolysis is done in ox and sheep blood agar. This point is extremely important. This is asked in MCQs, that hemolysis is done in which species blood agar. So that is ox and sheep blood agar. Also, now if we talk about hemolysis, so hemolysis is breakdown of RBC, destruction of RBC. So hemolysis, 4 types of hemolysis are produced by Staphylococcus aureus: alpha, beta, gamma, delta. Okay, 4 types of hemolysis. So there is a point in hemolysis only, double zone of hemolysis. So this point is extremely important. It is defined as the presence of 2 completely different zones of hemolysis around the staph colonies. Okay, double zone of hemolysis defined as the presence of 2 completely different zones of hemolysis around staph colonies. Let us try to understand that with the help of this diagram. So 2 zones are alpha hemolysin and beta hemolysin. So because of alpha hemolysin because of alpha toxin, beta hemolysin because of beta toxin. Now this alpha hemolysis is the zone of complete hemolysis. So if you see in this diagram, this is the zone of complete hemolysis, this is Staph colony, this is the zone of alpha hemolysis, and this is the zone of beta hemolysis. So in alpha hemolysis there is complete destruction of RBC, thus it appears transparent or completely white, and in this diagram, and the incomplete hemolysis is the wider one, and complete hemolysis is the narrow one. So this incomplete hemolysis is the area where only cell membrane of the RBC is damaged. Okay, so this is done. So these are the basic points. These are not important, but this table is important. I haven't completed this table, so I will just discuss it afterwards. But the next point is this: Coagulase positive and Coagulase negative Staphylococcus. So Coagulase positive and Coagulase negative. What is the meaning of Coagulase? Coagulase is an enzyme which is produced by the bacteria Staphylococcus species, and this Coagulase enzyme, what it does, it makes a wall of fibrin in the blood vessels which protect the Staphylococcus bacteria from the host immune system. Okay, so on that basis we have Coagulase positive, the Staphylococcus which makes Coagulase enzyme, and we have Coagulase negative, which is Staphylococcus which does not make Coagulase enzyme. So Coagulase positive, three most important examples are S. aureus, S. pseudointermedius, and S. ficus. Coagulase negative are epidermidis, S. epidermidis, S. saprophyticus, and S. chromosomes. Okay, so these are the most important example. And one important property of S. ficus is that it is non-hemolytic. It does not lead to the breakdown or destruction of RBCs. Okay, now this point is related to the exception. The generally Staphylococcus are catalase positive, as we have learned here. That Staphylococcus are catalase positive, but there are two exceptions: Staph aureus subspecies anaerobius and Staph sacrolyticus. These are catalase negative. We just have to learn this example. We don't have to go into any detail because books also does not contain any detail. Next is the diseases caused by Staphylococcus. So this is the most interesting topic of this chapter. Okay, so now let us start with the disease of Staphylococcus, that is bovine Staphylococcus mastitis. This is the first disease, and its causative agent is S. aureus. It can be acute, chronic, or subclinical. So acute means immediate onset, chronic means delayed onset, and subclinical means the clinical signs are not visible easily, or not visible at the surface level. Okay. Now, this Staphylococcus aureus in bovine Staphylococcus mastitis causes tissue necrosis in the udder with the help of a toxin called alpha hemolysin. Okay, so this is the effect of alpha hemolysis, that is, it causes tissue necrosis in the udder. And causative agent is S. aureus, and this is the method of action of. So this is the method of action of this Staphylococcus. So this is the blood vessel. In blood vessel there are molecules like fibrinogen and fibronectin. So what it will do is Staphylococcus, it contains some protein called Staphylococcus cell wall proteins, and these Staphylococcus cell wall protein will bind to fibrinogen, and after binding with fibrinogen they will help attach Staphylococcus to the wall of blood vessel, and there it will cause necrosis. So this is how the bovine Staphylococcus mastitis takes place or happens. Next is tick pyemia. So tick pyemia, its causative agent is again S. aureus, and it is an infection of lambs. It is found in the hilly grazing regions of Britain and Ireland, and its vector is tick, that is Ixodes ricinus. So this Ixodes ricinus insect or tick, it is the vector for tick pyemia. Okay, now lambs carry S. aureus on their skin and mucous membrane. Okay, so as we have learned that the S. aureus are found, or Staphylococcus in general are found as commensals of skin and mucous membrane. So lambs carry S. aureus on their skin and mucous membrane, and throat tick bites to the skin causes the S. aureus to enter into the body and cause tick pyemia. Okay, so they are normally present, these bacteria are normally present on the skin of lambs, but due to any trauma or injury on the skin of lambs, or due to the biting of this Ixodes ricinus tick, so these bacteria S. aureus are able to enter the body, and this then cause the infection. Now this Ixodes ricinus is also carrier or vector for another bacteria which is called Anaplasma phagocytophilum. So this Anaplasma phagocytophilum, it does not cause direct infection, it causes immunosuppression in lambs. It suppresses their immunity so that S. aureus is able to function more properly. Okay. Now what are the signs and symptoms of tick pyemia? So septicemia and rapid death. So septicemia, it is the compounded clinical state in which there is the presence of large number of microbes in the blood together with hyperthermia and toxemia. Okay, so that is septicemia. Second is localized abscess formation. So due to septicemia, there will be rapid death. Localized abscess formation that is pus containing cavities. Arthritis, that is inflammation of joint. Posterior paralysis. Posterior paralysis means that paralysis means partial paralysis. So due to the abscesses formation in the spinal cord, the posterior part of the body is not, the spinal cord is not able to control the posterior part of the body along with brain. So due to which the posterior part of the body undergoes partial paralysis or paralysis. And ill thrift. Ill thrift means generalized poor development of the animal. Okay. Now diagnosis. So diagnosis is always of two types, indicative and confirmatory. So if we talk about indicative diagnosis, then indicative diagnosis is indicative means we would just want to indicate that this lamb might have tick piemia infection. So we can just say that, or we can just confirm, or we can just say that that lamb was grazing on the hilly region of Britain and Ireland. So in this way we can say that yes, this lamb might have the infection of tick piemia, but this is the confirmatory diagnosis, and it is the best method to confirm or diagnose whether the organism, that animal, is suffering from that infection or not. So that is isolation of Asorius from pus. Obviously when we will be able to isolate Asorius, then we can easily say that this lamb is suffering from tick piemia. Indeed it is suffering. Okay, now treatment and control. So prophylactic treatment with antibiotics can be initiated at 1 week of age. For example, long-lasting tetracycline. So what is prophylactic treatment? Prophylactic treatment means preventive treatment. Preventive treatment means not the curative one but preventive one. Okay. Second method is dipping. So dipping is actually a method in which the small animals are dipped in the solution containing tick killing animals, and large animals are sprayed with these solutions, so these ticks are then killed. Okay. Third disease is exudative epidermatitis or greasy pig disease. So obviously this takes place in swine species of pig. So it is caused by S. hicus, and we learned two most important properties of S. hicus, that is Coagulase positive and non-hemolytic. So this exudative epidermatitis is a disease of sucklers and weaned pigs of 3 months of age. Suckler means the piglets which are completely dependent on their mother's milk, and weaned pig, that is the pigs which we have taken away from the mother's milk diet. Okay, it is highly contagious, that is highly infectious. Excessive sebaceous secretion. So these are the signs and symptoms. So highly contagious, excessive sebaceous secretion. We'll talk about that later. But these three, anorexic, depressed, febrile, these are the signs and symptoms of each and every disease. So if you are not able to remember any symptoms aside these three, so you will get some marks in the exam. Anorexic, depressed, febrile. Anorexic means low, no appetite. Depressed means low body function. And febrile means high body temperature, fever causing. Okay. So next is excessive sebaceous secretion. So this sebaceous secretion is non-pruritic type. So pruritic means non-itchy. Non-pruritic means non-itchy. So epidermatitis is greasy exudate. So this sebaceous gland, they become extra active due to this the infection of S. hicus, and they excessively secrete sebum or sebaceous secretions which are non-pruritic or non-itchy, and the skin of the pig is then covered by greasy exudate. Okay. Now rates, mortality rate is 90%, morbidity rate is 20 to 100%. Okay. So diagnosis. Confirmatory diagnosis, isolation of S. hicus from dermal lesions. Easy. We just have to isolate that particular bacteria from the lesion or the pus or any injury which is caused by, or any type of lesion which is caused by that bacteria. Okay. Now treatment and control. So isolation of affected pig. Obviously we have to isolate the effect. This treatment and control method is applicable for all disease. Isolation and control. Isolation of affected pigs. And second is, this is a novel method or new method, that is strains of S. hicus. Now there are two strains of S. hicus, virulent and avirulent. Actually every bacteria have two strains, virulent, avirulent. Okay, so this virulent, avirulent, so what we do in this method is we colonize the skin with avirulent strain. We will colonize the skin of the bacteria of the pig with avirulent strain, so that there is no space for virulent strain to act and cause infections. So this will prevent this colonization of skin by virulent strains of S. hicus. Okay. So next method for treatment and control of greasy pig or exudative epidermatitis is systemic antibiotic therapy with topical treatment. So what is topical treatment? Not tropical. This is topical. So topical means medicine applied to the skin. Any spray or cream which we are applying to the skin, that is called topical treatment. So using antiseptics and antibiotic therapy, systemic antibiotic, the antibiotic which acts on a particular system. So we will again give that topical treatment. This is also a method for treatment and control. Okay, so next disease is botulomycosis. So botulomycosis constitutes again the infamous SRES. Next, it is a chronic disease, that is, it has delayed onset. It will last for a very long time and it will progress slowly and slowly and slowly. It will develop slowly. Next is it is suppurative, that is pus forming. This bacteria is pus forming as aureus, so it will form pus and suppurative lesions will be formed. And this takes place only in two species, that is horses and sow. So if we talk about horses, then after castration due to infection in the spermatic cord. So in that case, in horses, after castration due to infection in spermatic cord, this will take place in horses, and in sow in their mammary gland. So means mature adult female pig. So in their mammary gland this will take place. So this is the general structure of the lesion caused in botryomycosis. So in botryomycosis, this is the animal. Suppose that this box is the animal and this is the lesion. So lesion is fibrous type. It is composed of fibrous tissue, and there is a foci of pus. There is centrally located pus in the center of this lesion. Okay. Last infection is staphylococcal infections in dogs and cats. So again, causative agent is Pseudo intermedius, Staphylococcus Pseudo intermedius, and sometimes S. aureus. So these are all the 6 signs or symptom, or we can say infections caused in dogs and cats due to Staphylococcus. So these are, first is pyoderma. Pyoderma is skin infection with pus formation. Okay. Next is otitis externa, that is inflammation of external ear. So external ear, I forgot to write here external, but inflammation of external ear. External ear means that is external auditory canal and pinna. Next is mastitis, that is inflammation of mammary gland. Endometritis, inflammation of the endometrium of uterus. Cystitis, inflammation of urinary bladder. And osteomyelitis, inflammation of bone or bone marrow. So these are all the 6 infections which are caused by Staphylococcus in dogs and cats. So this was all about Staphylococcus. Thank you.